If you've ever had this feeling that something was terribly wrong with the world that we live in, but you couldn't figure out just what it was, then you've come to the right place. Secret societies, mystery religions, and the Illuminati have been controlling our reality since the beginning of time. But not anymore, because there is an awakening happening, and you are about to become a part of it. Wake up, because this is a call for an uprising.
This is a call for an uprising. Welcome to today's show. As you can see, this is just going to be more ridiculousness involving Bill Gates, involving these patents, involving the names of these things that they're working with, all showing us that these people are nothing but Satanists. But before I get to that, I want to tell you guys about this plandemic video that's gone semi-viral that everybody has tried to watch, which YouTube has taken down over and over and over. I wanted to upload it to see what everyone thinks. I don't agree 100% with everything in it. Again, I don't trust a lot of these doctors who come on here. Although the things that the woman says are, to me, seem completely true. They make a lot of sense. I try not to side with people I don't know or can speak with directly. But I wanted to put it up so you could see it because they're censoring it for a reason. And you could see all this disinformation that's out there, this deflecting that's out there, attacking this video, not allowing to have a difference of expression or difference of opinion on this matter. I actually tried to upload it for you here on YouTube, and I put it under unlisted because that's how I upload my videos. I unlist them first so I could put in a thumbnail and all the other stuff, name the video. And in under a minute, the algorithm read this video, saw this video, which I guess has been fed to the algorithm, and they've been told anytime this comes up to delete it immediately. They deleted an unlisted video in under a minute and said it was dangerous content, harmful content. So it just shows you how badly they don't want this out there. So I wasn't able to get up here. I'm going to keep trying, but the video is available on my website, acallforanuprising.com. You can go there and check it out if you're a member. If you're not, consider becoming a member because there's no censorship over there. You could talk about these things and you could share information and interact with each other and talk about these things. So check it out over there. That's where it'll be for now. And I'll continue to try and upload it here just so you can have a chance to download it or look at it yourself, because I know everybody's been trying to at least just see it, and YouTube is not even allowing it to last up for a few seconds. I was able to grab it. Somebody uploaded it. I was able to grab it really quickly before it got taken down and download it. So that's how I was able to acquire the clip. Now onto the video here. I've been talking about the quantum dot. We've been all talking about the micro needle, the vax that they're going to be giving at some point in time that Bill Gates is behind, that they've patented. Okay, this is 060606. All of this stuff that's just right in our faces. And I know people go, well, this is a joke, right? That they're trying to, they're actually calling these things this thing and they're not hiding it. Well, I believe that God would never hide or allow these things to be hidden. I also believe, for everything I've studied on the occult and Satanists, that they need to show these things because that's part of how their sigil magic garbage works and enhances the power of their craft and all this crap that they believe in. Well, get a load of this. The enzyme that is supposed to be with the quantum microneedle, I'm sure a lot of you heard about it because it pretty much went viral the last couple of days, but thanks to people on my website, I was able to see this. Luciferase. That's right, Luciferase is the name of it. Luciferase, a genetic term for the class of oxidative enzymes that produce bioluminescence and is usually distinguished from a photoprotein. The name was first used by Raphael Dubos, who invented the words luciferin and luciferase for the substrate and enzyme respectively. Both words are derived from the Latin word Lucifer, meaning light bringer, obviously. And when you see the clips that I'm going to show you here of what I'm talking about, what they plan on doing through the vax that they're going to make mandatory, they love to use the words illuminate, light bearer, and Lucifer, right? But those are all coincidences. Let's all stick our arms out and let them put the shot in us. I mean, these people who don't believe are so dumb. I mean, they really are. Everything is right in front of us. They literally, and it's what I guess they call them, what, Generation Y? Is that the younger generation now? The dumbest generation. I shouldn't even say millennials because, yes, they're, a little bit slow. But the generation below them, I see these people, I see the videos they do. These people are so dumb. They think that they're literally trolling us with these names and these things. I mean, there's no hope for these people. They're, unfortunately, most of these kids who are being raised in this prideful society are going to likely be heading right to hell. But that's, it's all a big joke to them. We'll see how big of a joke it is when the trumpets sound. But the Luciferase are widely used in biotech for microscopy and as a reporter gene for many of the same applications as fluorescent proteins. It says, however, unlike fluorescent proteins, Luciferase do not require an external light source, but do require the addition of luciferin, the consumable substrate. Now, I know people are listening to this, they go, I don't even know what you're talking about. Completely understood. I mean, look, none of us use these scientific terms. There's people on the internet who will try to explain this who are full of crap anyway. They don't even know what these people are talking about. And most people just go, yeah, it sounds scientific. There's big words. I'll take the shot because science is real and it works. Right? And that's what they end up doing. So Bill Gates is building what they're calling the human implantable quantum dot, which is a microneedle vax, and it needs the enzyme called luciferase in order to make it work. And with each, the articles out there that are talk about it, reference the book of Revelation. They reference Daniel, even in Daniel chapter 12, verses 9 and 10. And he said, go the way, Daniel, for the words are closed up and sealed till the time of the end. Many shall be purified and made white and tried, but the wicked shall do wickedly. And none of the wicked shall understand, but the wise shall understand. And we're the wise who are understanding. So take a look as they try to explain this nonsense. But understand that without the Luciferase, the microneedle vaccine, the thing that's going to come here to be the saving grace for an outbreak that isn't even happening, is all going to tie in with the mark of the beast.
A chemical reaction that produces light within an organism is called bioluminescence. There's a wide variety of luminescent species, including some comb jellies, bacteria, dinoflagellates, fungi, and of course, the firefly. Organisms may use bioluminescence as a defense against predators, as a way to catch prey, or to attract a mate. But how exactly are they producing light? Fireflies express a protein called luciferase, which catalyzes a light-producing reaction. The reaction of luciferase takes place in two general steps. First is the adenylation of luciferin using ATP, and second is an oxidative decarboxylation of the luciferyl adenylate to form an electronically excited oxyluciferin. When the excited oxyluciferin moves back to the ground state, energy is released in the form of light. Although the exact mechanism is not known, the generally accepted mechanism is as follows. Luciferase first binds luciferin and ATP. Magnesium is needed to shield the negative charges on ATP. Luciferin acts as a nucleophile and attacks ATP to form a luciferyl adenylate still bound to the enzyme. Pyrophosphate is released. A residue in the active site of the enzyme then deprotonates the adenylate. The carb anion can now attack molecular oxygen. A ring is formed and AMP is released. This results in a high-energy dioxyethanone intermediate. The ring spontaneously breaks and we are left with electronically excited oxyluciferin and carbon dioxide. When oxyluciferin returns to the ground state, energy is released in the form of a photon of light. The excited oxyluciferin can tautomerize into the keto or enol form. Some scientists suggest that these two forms emit different wavelengths of light. Now let's take a look at the structure of firefly luciferase. This luciferase is composed of a single polypeptide chain with residues 4 through 436 making up the large N-terminal domain, and residues 440 through 544 making up the smaller C-terminal domain. The N-terminal domain contains a beta barrel and a 5-layered alpha-beta-alpha-beta-alpha structure. The C-terminal domain has 5 beta strands and 3 alpha helices. A disordered loop connects the two domains. Scientists are not sure exactly how the structure of luciferase allows it to function. They compared this structure to enzymes that catalyze similar reactions, like fatty acid coenzyme A ligase. Certain residues seem to be conserved on parts of the N and C terminal domains. This suggests that the active center is formed when the two domains come together upon substrate binding. This crystal structure of Japanese firefly luciferase is complex with a high-energy intermediate analog. You can see how the two domains came together around the substrate. People have incorporated luciferase into biotechnology and laboratory procedures. Next-gen pyrosequencing of DNA uses luciferase to produce flashes of light that correspond to the addition of nucleotides. dNTPs are added one at a time, and when the correct nucleotide is added to the template strand, pyrophosphate is released. This pyrophosphate is converted to ATP using the enzyme sulfurylase. The luciferase and luciferin in the medium can react with the ATP and a little flash of light is produced. By recording millions of flashes of light, a computer can sequence an entire genome. Luciferase can also be used in a reporter gene assay to study the expression of genes at a transcriptional level. The gene of interest is cloned slightly upstream of the luciferase gene in a vector. The vector DNA is inserted into cells, the cells grow and are then broken open to release all of the proteins. A luminometer is used to measure light emission, which directly relates to luciferase activity and the activity of your gene of interest.
The dual luciferase reporter assay system is an efficient method for performing dual reporter assays. With a dual reporter assay, a primary reporter of interest can be normalized against a control, allowing increased precision. This assay allows you to quickly quantify two luciferase signals from a single sample. Before you begin, make sure you have phosphate-buffered saline, a luminometer, and a siliconized polypropylene tube or small glass vial. Prepare the passive lysis buffer by adding 1 volume of 5x passive lysis buffer to 4 volumes of distilled water. Mix well and store at 4 degrees Celsius for up to but no longer than 1 month. Prepare luciferase assay reagent 2 by resuspending the lyophilized luciferase assay substrate in 10 milliliters of luciferase assay buffer 2. This reagent is stable for 1 month at minus 20 degrees Celsius or for 1 year at minus 70 degrees Celsius. Repeated freezing and thawing of this reagent may decrease its performance, so we recommend luciferase assay reagent 2 be dispensed into aliquots for each experimental use if the supplied volume is not used entirely. Prepare an adequate volume of stop-and-glow reagent to perform the desired number of DLR assays, 100 microliters of reagent per assay. Add 1 volume of 50x stop-and-glow substrate to 50 volumes of stop-and-glow buffer in a glass or siliconized polypropylene tube. Stop-and-glow reagent is best when prepared just before use. If stored at 22 degrees Celsius for 48 hours, the reagent's activity decreases by 8%. There are 2 methods for cell lysis, one recommended for passive lysis of cells in multi-well plates, and the other intended for active lysis by scraping adherent cells grown in culture dishes. Remove the growth medium from the cultured cells and gently apply a sufficient volume of phosphate-buffered saline to wash the surface of the culture vessel. Swirl the plate to remove detached cells and residual growth medium. Completely remove the rinse solution before applying passive lysis buffer reagent. Dispense into each culture well a minimum volume of passive lysis buffer to completely cover the cell monolayer. Place the culture plates on a rocking platform or orbital shaker with gentle motion to ensure complete and even coverage with 1x passive lysis buffer. Allow the plates to rock at room temperature for 15 minutes. Transfer the lysate to a tube or vial for further handling and storage. Alternatively, reporter assays may be performed directly in the plate. Because of the volumes added, this would typically only be in a 96-well plate. For active lysis, begin by removing the growth medium, then apply a sufficient volume of phosphate-buffered saline to rinse the bottom of the culture vessel. Swirl the vessel briefly to remove detached cells and residual growth medium. Completely remove the rinse solution from the culture vessel. Apply a sufficient volume of 1x passive lysis buffer to the cell layer in the culture vessel. Cells may be harvested immediately following the addition of passive lysis buffer by scraping vigorously with a disposable cell lifter or a rubber policeman. Pipette the accumulated lysate several times to obtain a homogeneous suspension. Transfer the lysate to a tube or vial for further handling and storage. Carefully transfer up to 20 microliters of cell lysate into a luminometer tube or multi-well plate. Place the plate or tube in the luminometer. Make sure you have placed the appropriate assay reagents in the luminometer and prep the injectors according to the instrument manual instructions. Run the protocol on the luminometer. A dual injector luminometer will add 100 microliters of Luciferase assay reagent 2, take a reading, and then add 100 microliters of stop and glow reagent and take a second reading. Remove the multi-well plate once all readings have been completed. For a single tube luminometer, insert subsequent samples until all readings are complete.